Hi, good afternoon. Very happy to be here. Thanks for the invitation. I need to start though with a little disclaimer. I'm not a cancer biologist nor a clinical scientist. But I still really value this opportunity to present you some of our work because I'm hoping it can bring us to think a little bit in a different direction and maybe start some new collaboration that we would be very open to. So if we don't spend our days talking about cancer, what do we think about? We think about mitochondria-nuclear communication. So a very basic aspect of cell biology that is central to the maintenance of cellular and molecular homeostasis. And that is because the communication between the mitochondria and the nucleus is required constantly through our lifetime for the coordinated expression of the mitochondrial genome and the nuclear genome. And this is required not only for making new mitochondria whenever that is needed, but also for global reprogramming of gene expression of the cells in order to adapt to stressful conditions, changes in nutrients availability, things that are sensed at the mitochondrial level. So what does this have to do with cancer? I would like to share with you a couple of different project ideas that we are thinking about. The first one is really based on work that has been done here by Gerald and other people in the field that have shown that exosomes that are released by adipose tissue cells that are energy stressed. So you can think of obese patients, diabetic patients. Those exosomes have a deleterious impact on cancer cell growth and metastasis. So the question that a student in the lab, Julia, is asking is a very simple one, is what drives these, the changes in the exosome cargo? If we compare obese and lean patient, the cargo is different, but what drives these changes? And we are thinking about that from the point of view of adipose tissue physiology. So the idea is that adipose tissue cells change their secretome, not because they know that there is a cancer around, they would change it no matter what, based on the obese phenotype. And the reason, and the model we are proposing is one where the metabolic stress that is induced by changes in nutrients availability and lipid overload is acting through mitochondria-nuclear communication to favor the expansion of the adipose tissue with deleterious impact on cancer growth. And so I'm not going through the approaches that she's taking or really the mechanism we are looking at, but the take-home message that I want to share with you is that really dissecting the mechanism of mitochondria-nuclear communication may be opening new avenues for therapeutic opportunities in which we can block the formation of these deleterious exosomes at the source rather than trying to act upon inhibit their activity down the road. And then the second idea is something that a postdoc in the lab is trying to bring about, Sana Mitra. Her idea is that understanding the mechanism of mitochondria-nuclear communication may actually have a role in dealing with chemoresistance. So why is that? Chemotherapies induce cell death through mitochondria-associated pathway, but very often the mitochondria manage to adapt to these pro-apoptotic stimuli and become resistant. This can happen through a lot of different mechanisms highlighted here. No matter what the mechanism is, based on what I just told you a couple of minutes ago, you may appreciate the fact that mitochondria-nuclear communication is required for all of this because it requires reprogramming of mitochondrial gene expression. And so again, this is another situation in which understanding the mechanism of communication may open up the way for new therapeutic opportunities. Thank you.